Hello, everybody. Can you hear me well, no? Yes.
Yes.
So, multiple sequence alignment. This is something that people love this. When people think of bioinformatics, people think of this thing. Well, or something like this. So, why people do multiple sequence alignment? Well, basically because you want to find what are the important parts in your gene or protein. Because when we look with BLAST, for example, you get a lot of sequences. And essentially, you know that each subject is similar to the query, but it doesn't mean that the subjects are similar between them. Let's say, for example, this is a typical example. A baby is similar to the father and is also similar to the mother. That doesn't mean that the father and the mother are similar together. Okay? And you can have all kind of variability. You can have a query that is similar to many things and that many things are different between them. So the next question is like, well, if all these things are similar to the query, what do they have in common? And you hope to find something that is kind of defining the property that you are looking for. For example, if all are proteins encoding for the same chemical reaction, same metabolic reaction, you would like to think that that metabolic reaction is catabolized by a particular site, the active site. And you will like to see that there is a kind of motif that characterizes that. So something that is conserved in all the proteins that do the same thing. Eventually, for example, you have, if you're a very rich person, you can isolate proteins that you know are doing something. Like you make an experiment where you find the proteins active when they are in the active site, and then you sequence the proteins that you explicitly know are doing something. Well, the other reason that people do this is to build, what is the name, this phylogenetic trees, no, kind of find evolution, which is an even harder problem that we are going to talk next week. Okay, we are not going to talk a lot today about trees today, just a little. But there is the general idea that by intuition makes sense that if you put all the sequence together and you check them and put the gaps in the correct place and you see what are the conserved columns, you will see, aha, what are the important parts, what are the essential parts that are conserved in all the proteins that do the same thing, and what is the particular thing that each one has different. Essentially, what makes you a human, let's say, or a primate, if you compare human with other apes, you see that we have more or less the same shape of the face, the hands and fingers and things like that. But some apes have tail, some don't have tail. So that is not essential. What defines a primate is conserved, all the attributes are conserved. Maybe apes don't have tail, but let's say fur, hairy skin. We tend to be less hairy. Of all the great apes like chimpanzee and gorilla, we humans are the less hairy ones. No, it's a good question. Why? Very good question. There is a book that I can recommend. It's a kind of old, not necessarily up to date, but it's a very relevant book called The Naked Ape. If you have the chance, it's super interesting. What else? So in general, people would like to do multiple alignment, and it's kind of what the biologists dream. Let's take all the sequences and we align them. And so far we did something similar. We did pairwise alignment. No, we did. Maybe I didn't tell you. This is the time I tell you the official name for global alignment is the method that we use to do global alignment is called Needleman-Wunsch. It's in the bibliography in the web page. There is a bibliography of all the relevant papers. This is one of the relevant. It's from 1970 or so. And they essentially explain how to do global alignment. And semi-global is kind of an extension of that. And then Smith and Waterman is the one used for local alignment, and it's from 78 or 79. So it took some time for people to go from one to the other. I mean, when you do it today, it kind of it's obvious, but it took very smart people many years to find the way to go from local to global alignment. And the other thing that we discussed, and this is an important point, that all these things required to make what we are calling the dot plot matrix. Essentially, initially we had dot plot with ones and zero, but then we put something like a score inside. And building that matrix requires a lot of memory and a lot of time. That is essentially in the order of m times n, where m is the size of the query and n is the length of the subject. Okay? That is important that you agree. And I mean, it's important that you tell me if you don't agree. Fine? Then, BLAST as a tool is only for database search. It's important to know that BLAST, even if called local alignment, is also search. It is not directly. This is kind of one of the texts is not following. Maybe I can make the slide. Because I changed the side of the slide. Okay, maybe. No. Whatever. This side. No. Well, I have to fix this one. Anyway, the important is that the cost is, again, you have d entries, so you have to do m times n for each one of the entries. You have m times n times d if you are doing it without an index, and BLAST is faster because they use an index and they save this part. So the number of entries that you test are much less, and then you still have to align them. You have to do the m times n or something like that. A dot plot around the seed. Okay, seed is called when you look in the index and you find it somewhere in the subject, that place that you find is technically called the seed, because it's where all the things grow from that place. Okay, any question? And you also should know that we fill the dot plot matrix with a score. We have different ways of score, let's say PAM or Blossom or something like that. That here I am represented with a function that is called score under 2 because it takes 2 inputs. And essentially you take the first sequence in the position I and the second sequence in position J, and you put that in the matrix IJ. Okay, the thing that we did in Excel, that we put the query in the rows and the subject in the columns, and we check the letters, and we fill according to the letter in the row and the letter in the column. Yes, no? Say something.
Yes.
Okay, or move the head. Okay. Yeah, okay, I have Arda, Yaya, and Ramal, and Turkan, of course, with the camera, so they can move the head. The rest have to speak. I say like Betul, Denise, Usan, Irem, Asle, they have to speak. Even Sarah is kind of, she has a picture. Anyway, okay, if you put the camera, you can, morning, you can move the head. And what happened? Okay, Atakan is not showing here anymore. Anyway. So it's maybe too early. So how we go from this? This is how we do a two sequence alignment, a pairwise alignment. How we go from pair from 2 to 3, essentially. Essentially, you have to think that if to align 2, you have to make a rectangle or a square. For make 3, you have to do in 3 dimensions. You have to fill a matrix, not a matrix, but a 3-dimensional thing. You essentially put in one side the first sequence, in the other the second, and in the other, and the third one 3 sequences. So it will be a cube, or technically a parallelepiped, but a cube. I don't have a cube here. We have to take my cubes. But you got the idea, no? You understand why it has to be a cube. Move your head, say something. Okay. Arda, you agree that it has to be a cube?
I don't understand why it has to be a cube.
Ah, okay. Why didn't you ask? Cube. Do we have a cube? No, I don't have my cube here. Okay, but here I have something like a cube. Remember how we did the alignment? And this will be my alignment. We put one sequence in the rows, one sequence in the columns, and we have to fill the points in the middle with the score, and then trying to find a diagonal. Let's say if you are doing global alignment, you try to find what is the way to go from one corner to the other corner, trying to maximize the scores that you win. You can think a game that you put positive and negative number here, and you have to go from one corner to the other, trying to collect the maximum number of points. But for doing that, you first have to fill the matrix with the scores. Okay. Now, if you have three values, you need to do it for the three. So you put one in one side, the other in the second, and if you have three, you have to put them in the other side, no? This one can be they can be different sizes, but because essentially you have to compare the first with the second, the first with the third, and the second with the third, and you have to put yourself in a combination, so you will essentially fill. This is a graphical representation, but essentially in the computer you have to fill a structure that corresponds in our mind to something like this, and you have to fill everything inside with a function that we will describe later called score3, a function that takes 3 amino acids, let's say, and tell you what is the substitution cost of putting these 3 amino acids together. If they are equal, it's good. If they are similar, it's not so bad. If they are very different, it's bad, and you will have positive and negative numbers according to some rule. Okay? Better? No? Okay? Yeah? Okay, move there. Good. Ramal is always serious, so yeah, go ahead.
We compare 3 amino acids simultaneously.
Again?
We compare 3 amino acids simultaneously in the same time.
Exactly. We have to compare because we are aligning 3 sequences, so we have to compare the 3 at the same time. Yeah, maybe I didn't say it explicitly, but the idea that we are going to take, yeah, I went very fast. I kind of jumped to conclusion that everybody knows what is a multiple sequence alignment. But essentially, yeah, we will have 3 or more amino acid sequences or nucleotide sequences, and we want to compare it all with all and try to find the best way to put gaps. If something that we should get from the previous class is that we can put gaps the way we like, but every time we put gaps, we get a different score. And since we have many alternatives, what is the best one? The best way to put gaps, it's called the optimal alignment, is the one that gives the maximal score. Okay, so ideally, I mean, if you are very, how can I say, if you have a lot of time, you put gaps in every position and you calculate the score, and then you put gaps in another position, you calculate the score. But the number of combinations is huge. So you better have a faster way. The faster way is the method that we say that you fill all the values and you try to find the diagonal. That is the shortcut. And that is much faster than testing everything. Okay? And that is why the idea of the diagonal is a powerful idea, because it's much faster than testing all the options. You remember that we say that the number of options is like something like 2 to the power of n or n plus 1, so it grows very fast. And in this case, it's just m times n, which is still big, but not so big as doing it case by case. Okay? What else? So the idea of aligning two things is to make a matrix. The idea of aligning three things is to make a cube, okay? And fill every coordinate I, J, K. You have three coordinates this time with the score that we need a function that we have to find this function that combines, that depends essentially on what is the amino acid in the first sequence and the second sequence and the third sequence. Agree? Okay, everybody agrees. Everybody moves. Or okay, at least nobody say no. Fine. What else? Then how this goes more in general. If you have three sequences, you essentially have to, no, but sorry, I didn't want to more general. So what is the cost of this? We say that we have to fill this matrix. What is the cost of filling this matrix? You have three sequences, let's say of length M1, M2, M3. Okay. I cannot call them M and N because I have three this time. And it will have many more. So I put a subindex now. So M is in general the length. M1 is the length of the first, M2 the length of the second. And to fill this thing is kind of essentially asking what is the volume of this. And the volume will be the length of one side, the length of the other, and the multiplication of these three things. And to simplify life even more, we will make a very strong simplification. I will say that all sequences have length M, which is not a bad assumption because in general, we will be aligning sequences that are more or less the same size. You know, since you are aligning many instances of the same gene, they tend to have them more or less the same size. Let's say around 100, around 1,000, something like that. Okay? So it is not a bad approximation. Give you an idea what's happening. But the bottom line is that if pairwise alignment was something that grow like M squared, aligning three sequences is something like M cube. Okay? And then how you go to the next step. That now we generalize. How we go to the next step? Tell me. Well, I am showing you the answer, but tell me how you go to the next step. Just one by one. How you align N sequences?
With a plot
Of? With a plot of?
N dimensions.
N
How many
dimensions.
lines we have?
Yeah. Do you know what is the name of a cube in N dimensions? No. My Star Trek friends are not here today. What is the name of a cube in N dimensions? In more than 3 dimensions? It's a hypercube. So yeah, we go to hyperdimensions. We are in the 4th dimension and 10 dimension and 100 dimension. And that is the thing that then you cannot see them, you cannot touch them. They are not more corresponding to a physical reality, but it's still something that our brain can imagine. We can essentially work in 2, 3, 4, 10, 200 dimensions. Is essentially the same thing. You just need to have more indices. So instead of having A, J, K, I have A1, A2, A3 until AN. And the score this time has N input. So score depends on the N values of the N amino acid from the sequence 1 in position I1, amino acid from sequence 2 in position I2, until amino acid of sequence N in position IN. Fine? Do you see how it goes more general? Ask questions now. Okay, Ramal has a question mark in the face. No? But there's no microphone. He has question, but not microphone. Oh my gosh. Yeah, okay. Okay, this is the easy part. So tell me if you don't understand so we can jump into the hard part. Usan, you are understanding? Yes. Yeah. Move your head in some way that says I don't. Yes or no?
Yes, yes. Understand.
Okay. Good. And I want to know what about Irem and Denise that don't speak. And okay, we are going to do something to make people talk. What we are going to do? Okay, and then second point, if doing for 2 sequence was filling a rectangle, doing for 3 was making a cube or a parallelepiped, and doing for 3 is a hypercube, for 4 is a hypercube, and so on, so on, so on. You realize that the volume of these things, the hypervolume, if you like, depends on the number of dimensions. So if everything is of size m, the cost will be m to the power of n, big N. Okay? Here, notice that I put n with big letter, with the capital, to make it different from the other n that I used before. And n here represent the number of sequences that we want to align. Okay? And the price, computational cost of this is m to the power of n. Okay? Fine? Okay.
Fine.
Ah, good. Good microphone. Good. I know what a Christmas gift for Raman will be microphone. So how much is that? Just to fix ideas. Tell me what does it. So let's say, for example, I'm thinking a gene like the 16S or maybe large protein. So just to fix ideas, I'm thinking that we are aligning sequence of 1,000 symbols. I mean, not a full genome, just one gene. It's kind of the average size of a gene in bacteria, at least. In bacteria, the average length of a gene is like 900, but let's say 1,000 is simpler. Okay? So the cost of aligning n sequences is 1,000 to the power of n, which is the same to, say, 10 to the power of 3 times n. Okay? Yes, no? Say yes. I cannot see you.
Yes.
Speak. Good. And especially say no if you don't agree. Okay? How many seconds? Now, we have to make some assumption here, and we later can see if this assumption makes sense or not. So how many seconds do we need to make this comparison? I am assuming that how many comparison we can do in 1 second. Well, that will depend, of course, on the computer and many things, but just to fix ideas, I am saying 1 million comparison in every second, 10 to the power of 6. So this is the number of comparisons, the number of cells that you have to fill, the number of time that you have to call the score function. That is essentially the number of time that you have to calculate score. And the number of time that you have to calculate the score is 10 to the power of 3 n's, but you can do 1 million of this every second. So the number of seconds is 10 to the power of 3n minus 6. Okay? Okay.
Why do we include 6 in the cost? It's just...
No, this is not the cost. This is the cost. This is the cost.
I assume
No, I'm asking
if
you.
there's an O, it's cost formula?
No, O essentially means that there is other factors, maybe a factor of 10, a factor of 1, a factor of, well, a factor of a constant multiplication. And you can even have some like smaller, lower powers. But the dominant part of the formula is the one that is inside. Essentially, what the O means that the formula is complex, but the most important part of the formula is this inside the O. Okay, so the O, it's a very good question. The O notation is used, the big O notation is essentially used to say, look, the formula is complex, but we are going to focus on the dominant part. We use it when we speak about cost, but it's not the only way to do it. I mean, it's not the only time that you use O. In general, you can use O, the O notation, every time you speak about things that grow fast and you want to know what is the growth rate. Okay. Oh my gosh.
Question.
Go ahead. It's just that, people normally have cats, but okay. This is surprising. Good. There are people who have better than cats. Good. Congratulations. Go ahead.
Okay, the slide before that, we said there is 1,000 comparison, and we said 10 times 3n. That's fine, I get that. But if we said, like next slide, there is 10,000 comparison, will we say again 10 times 3n? Not the 1 million, say the 1,000, and the two answers will be the same. Is it correct?
Okay. Let's go step by step, because I'm not completely sure if I understand you. But first point, I was careful to put the slide number. If you want, you can write the slide number to ask the question. So in the slide number 10, we say this. We say that if the sequence length is 1,000 letters, that is the length of the sequence of all the sequence that we want to align, then the cost, as we say, will be in the order of 1,000 to the power of n. Okay?
Yeah.
Then, that for simple math is the same. Writing this is exactly the same as writing this one. Okay?
Yeah.
Okay, that is the cost. That is the number of comparison that we have to do to align the n sequences of length 1,000. Okay? Then, how many seconds is that? Essentially, if we can make 1 million comparison in every second, we have to divide this number divided by 1 million. And 10 to the power of 3n divided by 1 million is the same as 10 to the power of 3n minus 6. This minus 6 is in the exponent. You have
okay.
10 to the power of 3n divided by 10 to the power of 6. When you divide, the exponent are divided. Okay?
I didn't think as we divide it to 1 million. We are asking
that is
if you are doing
like
1 million in 1 second, how many seconds it will take to do
exactly
10 times 3n?
Exactly. How many seconds? Essentially, you divide the number by 1 million. If it's 10 million, we will put minus 7. Okay?
Yeah, thank you.
No, good question. That is a good question. That is the way. Remember, I mean, come on, you are the old people here, not old, but experienced people. You know what is the goal of my courses. That the goal is that you ask questions.
This might be a silly question, but I was wondering why is the time unit is second here? Why do we not say minute?
No, I just started because okay, that is the kind of the next question, like how many that is in minutes or hours. Normally we take second because these things tend to be fast. It can be milliseconds, nanoseconds, or something. The unit that some unit of time that makes sense in the context. Yeah, it could have been milliseconds, or I mean, you know, in molecular biology, people always talk to me about nanogram by picoliter, no? Something, do you say like 10 nanogram by picoliter? So is it so? That the unit that people use when you pipette?
Microliter, usually microliter.
Microliter, okay. Microliter and nanogram or?
Yeah, in the gel.
Okay. Yeah, concentration, but the concentration, I think, is kind of, yeah. And in engineering, we don't think in nanogram or microliter and nanogram. We think about kilograms or tons and cubic meters, which are essentially, the proportions are the same. You essentially change the scale, but the proportions are the same. So in engineering, you work with cubic meters because you are making buildings. In molecular biology, you take milliliters because you are pipetting. But you use the unit that makes sense to you. I mean, you realize that Americans use different units for everything except seconds. The only thing that Americans agree with the rest of the world is that the time is measured in seconds. Okay, the rest, they are all crazy things. Okay.
Okay.
So how much is this? How much is this? If you have two sequences under this hypothesis, which later we can discuss if this hypothesis makes sense or not. Essentially, aligning two sequences take 1 second. Aligning 4 sequences take 1 million seconds. 8 sequences take, what is that? 10 to 18, 1 billion, 1 trillion, I don't know how to call them. And 12 seconds, 10 to 30. Okay? So, exercise. This is our quiz. I have a quiz that I have to open for you now. I will send in the chat, but in the meantime, I'm leaving you with this slide here, okay? Let me see, I will. And I'm going to, you can talk now, or if you don't want to talk, I will separate in several groups. Let me open a new. So this is the quiz of today. Since Ahmed didn't like seconds, okay, make it in minutes and days and years and so. Okay. He asked. So blame me. No, it was always the plan.
They're gonna blame you now.
Yeah, of course. But even if I prepared the quiz yesterday and I had the question already saved, okay, I'm sharing with you. No, it's not. No, what I mean is this, to be very honest, the question that you asked was a very good question, in my opinion. And I am so agreeing with that question that I asked the same question. Okay, that is also kind of funny. Like, I like the question because I asked it, but...
that sounds better.
Yeah.